Welcome to ASM Live at the 2014 American Society for Microbiology meeting in Boston, Massachusetts. We're here today to talk about some interesting, important, and sometimes very surprising stories of microbiology. We invite all of you in the studio audience to join on this conversation. If you have any questions, please go up to the mic and ask the questions. And all of you out there in the internet world, we invite you to send your questions at hashtag ASM2014. Please ask questions and join in on the conversation. This morning, we're going to be having a conversation about the question, is urine sterile? And we have with us Evann Hilt, Paul Schreckenberger, and Alan Wolfe. And I'm your host today, Stanley Malloy. I'm from San Diego State University, and our guests are all from Loyola University, Chicago. So this is an interesting question to be asking in 2014. People in clinical labs have been studying urine for so many years. How come people didn't realize that urine wasn't sterile before? Evan?
Well, they weren't looking before. It wasn't something that they were looking for. We were able to use a novel assay to be able to detect live bacteria in urine. For all these years, it's just whether or not a person had an infection. And with that, there was a certain threshold that they used to determine whether or not a person was suffering from infection or not. And all the bacteria that we see present are below that threshold. And
so what was the novel assay, and why did you even do that assay?
So we had, our group at Loyola had bacterial DNA evidence that we found with deep sequencing. And it wasn't, no one knew if that bacterial DNA represented live viable bacteria. And when you look at what's used in clinical microbiology labs with their standard urine culture, they only plate 1 microliter of urine aerobically for 24 hours. And a lot of the genera that we saw with this deep sequencing weren't culturable under those conditions. So we decided to expand the standard culture by plating 100 times more urine on various different medias and incubating in different conditions, such as 5% CO2 and anaerobic, and holding it for a longer period of time. And we were able to find the genera that we saw with deep sequencing under these conditions.
Stan, if we could just back up and go a little further backwards. We have a group of gynecologic surgeons at Loyola that are treating women with urinary tract disorders. A lot of them have incontinence, they have pelvic floor disorders. They require surgery, and a certain % of these patients develop post-surgical UTI infections, even though on day of surgery they were screened and were presumed negative. So the question that they were asking is, well, why are these women getting urinary tract infections when on the day of surgery their urine cultures reported no growth? And Dr. Wolfe at the time was doing some work with microbiome and sequencing, and he asked them to look at some of those, or he asked if he could look at some of those urines, and that's when he examined those urines on day of surgery and found the bacterial DNA. So then the question again was asked, well, if there's bacterial DNA in these urines, why is the lab calling them negative? And it's because the culture techniques that are standard of practice are based on the premise that to have a urinary tract infection, you have to have greater than 10 to the 5 bacteria in your urine. And so the culture conditions were set up to dilute out lower numbers of bacteria, presumably because they would have been urethral or vaginal contaminants. But in fact, what Evan has shown in her research is that urine is not sterile. And if you change the culture conditions and you include plating of higher amounts of urine and you vary the conditions of incubation, in fact, what's the latest number of % of patients that have, is it 75%?
No, it's 90% of urine cultures, urines that were deemed no growth by the standard clinical microbiology assay turn out to have bacteria, and some of them quite a large amount of different species of bacteria, but at relatively low numbers, well below that 10 to the 5, orders of magnitude below the 10 to the 5 CFUs per mil.
Well, some do and some don't. In most cases, it's the low numbers, but in some cases, we discovered bacteria present even at 10 to the 5. Mm-hmm. It's just that they didn't grow under the conditions that the lab typically used, because we presumed to know what the urinary pathogens were, E. coli, Proteus, Enterococcus, and the culture conditions that were set up to grow those organisms. They weren't set up to grow anaerobic bacteria or organisms that might take 2 days to grow or require CO2 incubation. Mm-hmm. So when Evann started, and you can describe a little bit all the different conditions you looked at, but what we found is when we altered the standard conditions, voila, urine is not sterile.
So Paul, from the clinical lab perspective, are people accepting this concept? Is this a concept that now is being thought about by clinical labs across the country and around the world?
It is not, because it's difficult to dispel old dogmas. We had a symposium at last year's ASM on this very topic, and it was very well attended. We've gotten a lot of feedback. We're publishing on this. We're trying to get the word out. This event today is helpful in that regard, not just to educate scientific audience, but the lay audience, that we have to think about urine differently. And by the way, the fact that it's not sterile could very well be a good thing, because as you know, the human microbiome does all the work for our body. And what the interesting part is not just the discovery that there's a urine microbiome, but what is it there for? Right. And what is its natural function? And what do antibiotics do to people to destroy that urine microbiome?
Yeah, we have statistically significant data that argues that there is a protective microbiome in the bladder. And so the question does become, should you give antibiotics or not? Because the antibiotics could kill off the protective microbiome and make individuals susceptible to invaders. So there's a question of whether or not there's a C. diff type of a scenario going on in the bladder. And the answer, of course, is be patient. We're working this out. We don't know who the good guys are, who the bad guys are. We have evidence of associations between, bacteria that we see more often in individuals with no symptoms, and a large number of organisms that are highly associated with individuals with a urinary tract disorder.
Let me come back to something that you mentioned earlier. You said that you mentioned 90% of the people with a certain situation showed bacteria in the urine. But do your DNA sequencing results indicate that virtually everyone has bacteria in the urine? If this is a normal microbiome that has a protective effect, you would think that you would find this in everyone. Is that correct?
Let me take that one. Urine is a low biomass environment. It's not like the gut where there's 10 to a gazillion numbers of bacteria. It's very small numbers in the thousands, hundreds of thousands. And when we sequence, what we find is about, depending upon the type of individual that we're sequencing, we get anywhere from 40% to 80%, let's say, of the urine samples that we can amplify by PCR and then sequence. But of the group that comes out as PCR and sequence negative, Evann's able to, about half of those, Evann's able to isolate bacteria. So then what we are left with is maybe about 25% of all the urine samples that we have no evidence of bacteria. But we don't believe that those 25% don't have bacteria. There are technical reasons why we might not be able to sequence, and certainly if they've got an organism that our growth conditions don't capture, we're not going to isolate them. So at this point, we would argue that probably everybody has bacteria in their bladder. And of course, the question now is what are all those bacteria doing?
Mm-hmm.
So, you know, we've got many different thoughts, of course. As soon as you make a discovery, you start to wonder about what it means. And, you know, one of the things you can think about is it's sort of the C. difficile story. Maybe those normal flora in the bladder are what protect the bladder and keep the invaders out. And it's when you lose your normal flora, when you become dysbiotic, that's when you're susceptible to those infections. You have to ask, why did only 10% of our patients undergoing surgery get post-surgical UTIs? Why do only 10% of people with Foley catheters get UTIs? Why do some women that get UTIs get them over and over and over? And so we wonder now about these things, and we think that the answer is in studying the urine microbiome, and perhaps the microbiome has been disturbed, and that's why certain people are more prone to these conditions than others.
We have a colleague who's an immunologist and cell biologist who's established, his name is Fong Li, and he's established urothelial cell, primary cell cultures, and from that, cell lines. And so one of the things that we're doing with Evann's bacteria is putting them together with the urothelial cells and asking how do they interact. And all of that work is really sort of preliminary at this point.
Very interesting. Before we go on, I'd like to remind people in the audience, both here in our studio and on the internet, please ask questions if any questions come up during the discussion. So I was just about to ask the question of how would you test cause and effect to see if some bacteria were beneficial? And it sounds like you're going to begin by looking at these cultured lines.
Mm-hmm. Yeah, so all the bacteria that I've been able to cultivate, we've saved them, and so we have a huge collection of urinary isolates, and that's one of the ways that we're going to start to see, you know, how they interact with the host and what factors they're secreting and how's the host responding to those factors, and also competing them with each other. So, you know, we see some that are beneficial or some that may be, you know, non-beneficial, and testing those interactions and seeing what's actually going on.
So let's
part of our plan is to develop a bank of organisms that come from these urines, because you really need the organisms to study any kind of models. I also thought maybe, you started mentioning it before, but maybe for the clinicians and clinical microbiologists that might be listening to this, you could explain briefly how you've modified the routine urine procedure to get these organisms to grow. Could you just kind of go through that briefly?
Yeah, so by expanding the culture conditions, we've plated 100 times more urine than what's normally done. We've also expanded onto different medias, including a chocolate agar as well as an anaerobic blood agar. We put it into aerobic conditions, 5% CO2, and anaerobic conditions, all at 35 degrees Celsius, and hold for 48 hours instead of the 24 hours that's normally used. So
That's great. Those are all conditions that could really be used in a clinical laboratory. We have a question from the audience.
Hey, so urine is actually a fairly poor growth medium. So I'm just curious about the absolute number of organisms that you think are going on, because you know you're isolating these and then enriching them. So if there's 1 to 2 bacteria, not so relevant. But if it's more like 10,000 or so, maybe it is. So do you have a handle on that?
not at this time. I know
You do have a handle on the number of
yeah.
organisms, the average quantities that you're growing with these expanded techniques.
Yeah, with the expanded technique, the average colony-forming units is about 300, so it is a low threshold. Moving forward, though, we definitely want to try to grow these organisms in maybe an artificial urine media to really get what environment they're actually living in instead of enriching the conditions. But we don't have too great of a handle on the actual environment yet.
And then,
Well, I'd like to add to that the answer. We're using urine as a reporter, right? You know, urine is what we can grab, but what we'd really like to know is which bacteria are associated with the tissue, right? And so even 50 bacteria associated with a cell, like a urothelial cell, could have a major impact, especially if they're inside, right? As your mentor has shown, they can be. So we're actually started doing that. We've taken, and Evann's been involved with this, taking urothelial cells that are shed into the urine and trying to grow bacteria from those shed urothelial cells. We also take biopsies from our urogynecologist colleagues led by Linda Brubaker and trying to culture bacteria and sequence as well from those biopsies. And we sometimes are able to, Evann's able to grow bacteria, we're able to sequence bacteria from some of those samples.
so in your suprapubic catheter specimens, did you take a look at the cell population that you had there microscopically and see if you can identify bacteria adherent to the urothelial cells?
We did. So you're referring to our earlier study where we took urine directly from the bladder to avoid vulvovaginal contamination. By the way, this is all of our work's been done with women. And so a suprapubic aspirate is to take a needle through the lower abdomen. And from those aspirate samples, we sequenced bacteria. So we knew what genera were present. And the more recent work has validated what we saw. We did do a little bit of microscopic work, you know, gram staining, for example, like the Clin Lab normally does. And we saw some evidence of bacteria that we thought would be alive. More recently, we've gotten IRB approval to go back and take some more suprapubic aspirates. And so we've begun to receive them. Evann, you've cultured some of those, haven't you?
Yeah, they match what we've been seeing throughout our work with the transurethral catheter specimens. So that's a pretty good indicator of what is actually in the bladder.
You know, the other thing that you alluded to is the quantity that's significant. And, you know, that's another myth. The fact that infections are present only when the bacteria are present at 10 to the 5 or greater. You know, that was never the intent of Cass's original report. And in fact, the amount of urine, the amount of bacteria present in a urine in people that have urinary tract infection varies throughout the 24-hour clock. When you get up in the morning and the urine's been concentrating during the night, sure, it can be 10 to the 5. But at 11 a.m., after you've had your coffee and you've urinated a few times, it can be 10 to the 2. And that's also significant. But laboratories are not culturing at 10 to the 2. And so I think that we miss a lot of real urinary tract infections by setting these cutoff limits based on another dogma that we think needs to be trashed. We basically have to relearn everything about urinary tract because we were misled. Our beliefs were unfounded. We're now, with the new science, are realizing that everything we've been taught is probably wrong. The clinicians that we work with are quite distraught over this. One of our clinician colleagues said, well, this is terrible. You took away my dogma, but you didn't give me anything to replace it.
We have another question from the audience. Please introduce yourself.
Yes, my name is Marie Peslow, and I'm from the University of California, Irvine. And my question is related to the difference in bacteria that you are seeing versus the normal flora that we normally see, or maybe don't see, from the vagina and the cervix. Are these organisms similar? Are they, and when you refer to bacteria, are you seeing mostly bacteria, or are fungi involved as well?
We haven't looked at any fungi. All of our work is with bacteria, but we do see similarities in the populations that we're culturing. So a large sample, if 25 to 50% of our women are lactobacillus dominant, so resembling the vagina. Another 25% are Gardnerella, and then we have our mix. But yeah, they are very similar. As for if they are the same communities, the same bacteria between the two sites, that's something that we're going to be doing more research on. We have a little bit of evidence from the earlier work with sequencing, but no culture data yet, and so that's something moving forward. But we do see similar communities.
And
I think we see some of the same organisms. I wouldn't necessarily go as far as to say they're similar communities. In our first paper that we published, we compared the sequencing results from suprapubic aspirates, protected catheters, and voided urines. And the suprapubic urines and the catheter-collected urines were quite similar, and they were different from what we saw in the voided urines, because the voided urines in women contain mixtures of vaginal and urine organisms. So there are some similarities, but there's a lot of differences. And I think the biggest surprise to me as a clinical microbiologist, Marie, is that what I call the A list, it's all the organisms that start with the letter A that we see in urine that we were not reporting in our clinical lab. Aerococcus, Actinobaculum, Aliscardovia, Actinomyces. We see these in a high level of frequency in all of our urines, both our symptomatic patients and our asymptomatic patients. And historically, when we look back at our records, we weren't reporting those on patients because they don't grow on McConkey in 24 hours in aerobic atmosphere. So I think that one of the things we're going to have to learn is what really are the urinary tract pathogens, because our understanding of what's a pathogen has changed. Another thing that came out of our first study, these guys will remember, is one of the urines that we had a patient, and they grew greater than 10 to the 5th E. coli. It was the only thing they grew out of their urine. When Alan sequenced it, the E. coli DNA in that specimen was less than 1% of the total bacterial DNA. 50% of the bacterial DNA in that sample was Aerococcus urinae, which the lab completely missed. So they're treating an E. coli urinary tract infection, as far as we knew, could have just been a fecal contaminant, not even a uropathogenic strain. So I think that everything we knew about urine, we have to question and relearn.
I think that's the most important point. Everything that we think we know has to be reassessed. It may be right, but also it may be wrong. And that's really what we've done here, is simply sort of throw everything up in the air and say, okay, now we know that there are bacteria there. So who's good, who's bad, how do they all interact with each other and the host? And that's the next step, is to figure out what's actually going on in the bladder.
I do have one more question, and that's related to the post-surgical UTIs that you mentioned. And now these patients have findings that are consistent with urinary tract infection, is that correct?
Correct.
Okay. And how about the asymptomatic patients? We know, especially in pregnant females, that there is the chance of asymptomatic bacteria, which can have a negative impact on the fetus. Are you looking at those as well?
We've put in a couple of applications for grant money to do that sort of work, asking questions about how does the urinary microbiome affect premature birth. We just haven't been able to get any money yet. We're definitely on it.
Yeah. Thank you.
We have a question from the internet, but before we get to that, I'll say that's one of the trouble with dogma, is that if the people on the grant panel believe the dogma, it's hard to get the grant money to dispel it.
But talking about dogma, often, Paul alluded to it, but dogma very often occurs because someone established something that was correct for one condition, and then for some reason, that test or that idea migrated to another scenario, it becomes rock solid. And then somebody else comes along and says, the dogma's not right, and they're shouted down. So we're not the first to show that there are bacteria in the bladder. And we're not the first to say that the threshold of 10 to the 5 CFUs per mil is the gold standard. In fact, Walter Stamm in the 1980s said that 10 to the 2 colony-forming units per mil of a known uropathogen is indicative of a lower urinary tract infection. The 10 to the 5 came from pyelonephritis. Ed Cass didn't want to do surgery on people, kidney surgery on people who had pyelonephritis. So that he established that 10 to the 5 for that purpose and that purpose only. It migrated to urinary tract infections. And Walter Stamm said 10 to the 2 is the threshold. He was ignored. And a woman by the name of Rosalind Maskell in the UK took suprapubic aspirates and was able from those to grow lactobacillus and Gardnerella, and she was shouted down. And so we know about those results. So we had a pretty good idea that when we went to looking, that we would find the difference that we had is that we had all this deep sequencing to back us up, and a community was willing to believe that microorganisms were everywhere.
We have another question from the internet.
Yes, Chris Longshaw from Twitter has a question regarding the previous discussion that a bladder dysbiosis could be linked to disease, and is wondering, is there a role for a microflora transplant for UTIs?
Mm-hmm.
I can answer that question. Linda Brubaker and I have been talking about doing urinary microbiome transplants. There are some older women who have really serious recurrent chronic problems. These women would be willing to do almost anything to solve their problems. So we're not quite ready yet because we really don't know what a non-dysbiotic bladder looks like. But the moment we feel comfortable with that, we would like to try it on some of these extreme patients. We could completely change their world if it worked.
We have another question from our audience.
Hi, Patrick Olson from WashU in St. Louis. You mentioned briefly that all your data is from females. Do you have any idea of what the urinary biota might be in males, and how this fits in with the fact that males don't get urinary tract infections?
Yes, so we mainly focus on women because that's our patient population, but others out there, such as David Nelson and Dennis Fortenberry, they're doing work with the male urinary microbiome. And we do see some similarities between males and females.
Yes, so actually males do get UTIs. They just don't get them with the same frequency as women do. As Evan said, there are lots of similarities. There's a few papers out there. What they haven't done is to take a suprapubic aspirate. So we can't say that the male bladder has any bacteria. The lower urinary tract of males does. So that could just be concentrated in the distal urethra. But it might actually be that there are bacteria in the bladder.
So Dennis Fortenberry was on our symposium last year at the ASM, and he talked about a little bit about this. And one of the interesting things I found is you said that males don't have as high a frequency of UTI, but they do have STDs. And one of the STDs is chlamydia, and one of the important ingredients for chlamydia to metabolize is they need a source of indole. And so you can readily see where that might come from a female, where they have a vaginal microbiome, and some organisms are breaking down tryptophan to form indole. But where would a male get the indole? So that's why he started studying the male urethra, and guess what he found? One of the dominant organisms, which we don't see in the female, but we see in the male, or he sees in the male, is Propionibacterium acnes, an indole producer.
Right.
So we do know that males have it, like at least in the urethra, a microbiome, and it does contribute to, in some cases, enhancing infection. You know, the microbiome is an interesting thing. It works both ways. And if you disrupt it, those normal organisms can suddenly turn pathogenic. So I'd like to put a plug in for Martin Blazer's new book,
Right.
Missing Microbes. It's a fascinating read. It's only been out about a month. And I encourage everybody interested in this topic, actually I encourage everybody, even if you're not interested in this topic, to read his book. It'll blow your mind about how the microbiome works in our body and how just disrupting it through all the modern conveniences like antibiotics and hygiene and C-sections is really having a lot of long-term effects.
We're nearing the end, but we have a final question from our studio audience.
Hi, I'm Vijay Aswani from Marshfield, Wisconsin. I'm a pediatrician, so I'm disturbed by some of this. For one thing, we now screen a lot of adolescents for sexually transmitted diseases using PCR technology, specifically chlamydia. Is it possible that we are getting false positives and treating those, and as a result of undetected organisms? The second question I had was, did you find any variation in the microbiome based on ethnicity or sexual activity on the females in question?
So there is a real danger that we start to report that there are bacteria in a urine sample and that the clinicians would rush to treat it. Well, if you're treating you're treating a urine, an individual because there are bacteria there and there's bacteria protective, that would be a really the wrong approach. So that there's an issue about how do we inform and educate clinical microbiologists and clinicians as to what to do with the new information. Like I said, we don't really know yet what we should do, but the one thing we shouldn't do is to act like all bacteria are bad. And your second question was...
If there is a variation in the microbiome
right,
across
so
ethnicities and sexual activity.
so we have, we've not done those studies yet, but we're fully aware of the work that's been done by Jacques Ravel and Larry Forney and their colleagues on the vaginal microbiome, in which there is really, there are racial and ethnicity differences. We've also want to do studies on the stability of the urinary microbiome, taking the same individuals over time and sampling them multiple times and asking, how does the microbiome change? We have a little bit of data that Evann has, and there are some individuals where it's stable, some individuals where it varies widely. And that's what they saw in the vaginal microbiome, but the sample size is really tiny at this point.
So we should wrap this up. We've spent so much time talking about this amazing concept that urine is not sterile that we haven't talked about one of the other really important outcomes of this type of research, and that is your data suggesting that there's a strong correlation with certain kinds of microbes and overactive bladders in women. Right. So that's something everyone in the audience should look forward to hearing more about in the future. But it clearly points to the fact that this isn't simply a discovery of what exists out there, but it's a discovery that has very important clinical significance. And so I think that's an important point. Another thing I think we should really emphasize is that it would have been very, very difficult to ask these questions a decade or 2 decades ago without the advent of deep sequencing, which was the criteria that would help neutralize some of the naysayers in this type of business.
It's really hard to argue against that data because it's voluminous, right?
Right. Yeah. So it's a very exciting time in microbiology, and I think this emphasizes how vast the landscape is that we can probe in terms of microbial functions on us and in the world around us. So I'd like to thank you again before we finish, Paul Schreckenberger, Alan Wolfe, and Evann Hilt. Thank you very much, and thank all of you in the audience. Please come join us again for the next session of ASM Live.
Explore the fundamental role of microbes in the natural history of our planet with Microbes and Evolution, the world that Darwin never saw. Available at estore.asm.org.